when we first started Synberc here, we thought the idea of bringing these people together who had different expertise, all interested in this common theme, would be able to synergize and build upon each other.
It would have been very easy for me to come into Synberc and say, hey, I'm an electrical engineer, and for them to say, what are you doing here? We're not interested in this. You don't know enough. But they were like, this is a new viewpoint. You're bringing something to the table.
Unlike people who come from the straight biological perspective, who see the stochasticism and the messiness and the chaos of biology, and are interested in understanding it, engineers seem to me to be people who come from a world in which you can order things, and things can be named and parts numbered and ordered.
If I have a pathway and I have a problem, then I can collaborate with other PIs at Synberc in addressing the problem. You see those collaborations going all the time.
We are concerned with the process by which you make the raw DNA, the short piece of DNA, how you assemble those, how then you put them so that they work in bacterial or human cells.
So if you want to put together a large set of parts and you have a starting parts library, how do you put them together in the quickest and most efficient way possible? There's a whole pipeline of steps that must be taken.
We have a lot of issues connecting genetic elements together to create circuits, and some of that is due to errant transcription. We're hoping to make these circuits more robust and easier to combine together.
We've also expanded the tools so that we've made it easier for everybody to engineer biology, not just people who are within Synberc, and we're sharing those tools widely with the public.
So once you have this pipeline going from raw DNA to DNA that's working really well in cells, you have applications.
So we are part of what's called the Microbial Chemicals Factory testbed of Synberc, which means that what my lab does is to engineer microbes, bacteria mostly, for chemical production. Those are chemicals that may be useful for fuels, they could be used as commodity chemicals for materials, even pharmaceutical intermediates.
So if you're talking about tissue engineering, doing, basically regenerative medicine, it's important that your engineered device can function across thousands and thousands of cells. Engineering gene packaging is going to enable us to do that.
Often the task is not in building the pathway to make the biofuel, but it's in making higher quantities of biofuel more efficiently. So we like to develop the technologies that make those existing pathways work more efficiently.
Right now, when people build their own pathway, they lost all the regulatory system. And we are trying to reconstruct this regulatory system in a synthetic pathway.
By integrating computation, it will allow us to not only share and design systems, but then verify them, incorporate safety and best practice aspects as well.
Lots of people in Synberc, they develop tools, and I can use these tools to build my circuits. I can also answer questions like, for certain pathway, which is the best way to build the system and who can make your pathway work better.
People are getting data, they're getting statistics, they're looking at robustness, they're looking more at the system rather than a specific problem.